In today's lesson, our focus is going to be on how a cell can copy its DNA inside the nucleus so that as it splits, it has a version for each new cell. This process is called DNA replication. If you look at the photograph, what you'll notice is there's a couple words that we're going to emphasize in this short little video. It's also found on sections 12.2 of your textbooks. The first word is called RNA primase. And RNA, or excuse me, DNA primase. In DNA primase, along with DNA ligase and helicase or DNA helicase, these are the enzymes that are necessary. And I think of this as the tools that are required to make the cell operate. And so it's like the screwdrivers and the hammers, the really kind of the tools necessary to perform so the cell can grow and ready itself for the day-to-day operations and even for cell division. We'll also look at the other terms in here. There is some lagging strands, what's called the lagging strand and the leading strand, along with another term called the Okazaki fragment. So this is our focus. We're going to be using some new vocabulary, new terms that will be introduced, along with just the process of how it copies this. So this idea is that when a cell copies and can divide, is allowing this to replicate the information or the DNA during a process called interphase. Interphase is just this time of growth of a cell. And one of the areas that I want just to just mention here with this photograph is this area right here. This is referred to as a chromosome. And a chromosome, in this case, is this DNA that has a ladder-like structure. And this part is referred to as a monovalent chromosome. There is a second part that we're going to begin into, and this is how it copies itself. And when it does, in the completion, it will have two parts, a lagging strand and a leading strand. And these two parts are referred to as the bivalent portion of the chromosome. So mono meaning one, this one part, and then we're going to eventually produce two parts here called the bivalent portion of the chromosome. So the monovalent and the bivalent are areas of a chromosome or structure that has information on the DNA. So before we look at this, this is kind of an overall general view of the DNA molecule. Remember, it has in blue a parent strand. In other words, this is the original copy of A's, G's, T's, and C's. And then there's this area, it almost looks like a fork. And this is an area that's been opened up to allow these A's, G's, T's, and C's to be exposed and potentially find a new mate or a new complementary base. So there are certain steps that we'll highlight here, and I'd highly recommend just to write them down just to slow you down and think about things a little bit differently. Again, found also in 12.2 of your textbooks. But really, the first step, as I was suggesting, is just where the DNA unravels and unzips. In other words, it's a helical structure, and when it unravels and unzips, it exposes areas of the DNA. So an enzyme does this, and this enzyme is called DNA helicase. Again, it's very similar to the structure of DNA. It's a helical structure. And so one of the functionalities of DNA helicase is to unravel and unzip or untwist. When this happens, there are molecules called free-floating nucleotides, or we can just call nucleotides. And this consists of a phosphate, a sugar, and one of four bases. And those are just almost like the air in the room here. We don't know necessarily, we're not going to necessarily mention about where it comes from. It's just present. So you can take a giant leap of faith, but these nucleotides are present when this is happening. And what will occur is that those new free floating nucleotides will try to find a complementary base on the DNA molecule. So on the part that's been unzipped and exposed. And then a second enzyme that we'll highlight here is called DNA polymerase, will attach the two together, adds those bases together, and form these two identical strands, which again, in the earlier slide, we referred to those as the bivalent chromosomes. So it goes from a monovalent chromosome, in other words, just one helical structure, to now two helical structures. And these are really kind of the overall general steps, and it requires some tools or enzymes in order to do this. This is a little easier to see this happening. We have our untwisted molecule of DNA at this point, and the next, or the middle photograph, you can see that these free-floating nucleotides that I mentioned are going to go out and find a complement. Remember the base rule that G's will always form bonds with C's and vice versa, and T's will always find a bond with an A, and that's what you're seeing being formed here. So in purple, you're going to find the old strand, and then in the red, we're going to see the new strand being formed as a result of this unzipping action. Since they're identical, we use a term that helps describe this, and since we'll use family terms oftentimes in biology, we call these sister chromatids. These are identical. We're going to assume that there's no mistakes or errors that have occurred, and so we refer to these as sister chromatids, or in the whole, we'll just even can refer to it as just a chromosome. In steps 1 through 5, this is kind of spelled out step by step, and I'm not going to necessarily read the sentences. You can pause the video and you can kind of go through just as we had stated, but this is just another way of looking at the steps involved in more graphical, and also where some information is step by step associated with each diagram. All right, one of the things that I wanted to bring out that's a little bit different is replication, of course, is a little more complicated than what we just saw in those earlier photographs. So there are two separate strands of DNA, and we've mentioned earlier that one is referred to as the leading, and one is referred to as the lagging strand. And what that means is that when they're copied in different directions. And so imagine like one-way roads where you have direction flowing one way and the other side is flowing the opposite way. One side is more fluid than the other side. And so we're going to associate some terms or some characteristics from the leading side and the lagging side. Let's first of all start with the tool necessary for this. This is called DNA polymerase. DNA polymerase is one of the tools that's going to help build these molecules on both sides of this open DNA molecule, but it'll only travel in one direction, adding these nucleotides, and it's very, very specific. They call that a 5' to 3' end as far as this direction. And what I wanted to bring out here is that we'll start with the leading strand. This runs in the kind of the correct way. In other words, it's very fluid and it happens very quickly, one after another. So as a T is exposed, an A will combine to it, the next step, and it just keeps on moving forward. This is the leading strand, and it moves very quickly and continuously is probably the better word, continuously in a fashion that's just as fast as it's being unwound with the enzyme called DNA helicase. And you can see where this is exposed here. So this is my leading strand on this side, and you can see that we're going to form the complementary side just as fast as it gets unwound. The other side of this is referred to as the lagging strand. And the lagging strand, it doesn't run as efficiently, but it runs in the wrong way or the opposite way. Probably the best way of saying that is the opposite way. This area right up here where my red circle is referred to as the replication fork. This is where the helicase is unwinding the DNA and where it's beginning to be copied back behind it. Well, in the case of DNA helicase or polymerase, excuse me, it will go the opposite direction. But notice here is it will start here, for example, putting these free-floating nucleotides, just like the other side, together, but it will only do that in small little chunks before it gets dropped, and another chunk is going to be formed, and another one after that. And so it just does things in small little pieces. It's not complete like its counterpart on the leading strand. So this new DNA polymerase attaches, moves forward, and again, it just has these small little chunks that it's putting together. It copies it until it reaches the previous section, then it falls off, and you can understand where it just has some linkages that need to be come together in order for it to be complete. And what's seen in the arrow here is some of that linkage that's occurring with another enzyme called DNA ligase. And so ligase is responsible for linking the chunks or the sections together, um, and to make it as continuous as the leading strand. So we get those chunks all put together, and the positive part is it will look just like the other side. Now, a term that's come out based on the individual first noticed this, his Dr. Okazaki, he was an American scientist from California. He noticed this and the difference between the two sides. And so today we still oftentimes refer to these fragments or pieces on the lagging strand as Okazaki fragments. Your book labels it that way, and most videos will also make reference to it, so I will as well. So one of the things that you can see on this diagram here is all the enzymes at work. You have to visualize how it's working and operating. One of the first ones, remember, is this DNA helicase that will come by and unravel or unzip the DNA molecule from a helical form, exposing both new sides here at this site called the replication fork. Now, on one side, DNA is going to go continuously from this 5' to 3' end, and it's going to do that with DNA polymerase in a continuous cycle. On the other end, it happens as well, but it just happens in small little chunks called the lagging strand. DNA polymerase will do the same thing, but it will fall off, will continue on right here, fall off. And what's necessary is we need another tool or lagging strand on this lagging strand called DNA ligase to link them all together. When that happens, we'll get two um, continuous replicas of the original strand. So here is one side that has a um, initial side that was the original in blue, and then the new side in red. Same thing over here. We have one side that was the original, and then one on the side that is the new side. Now, what you'll see in your textbooks, they'll refer to as this copying piece as something that's referenced as semi-conservative. In other words, it has a little bit of the new and has a little bit of the old. Semi-conservative replication. And that's with all of those enzymes working together to form this complete strand. Now, as far as the accuracy and sometimes even the repair of DNA, it's very, very precise. It's estimated that there is one error in every about 10,000 paired nucleotides. In other words, it doesn't happen very often. I know when I'm typing, I make an error, it seems like every 10 words. Um, this is extremely accurate, and we call these changes a mutation. Now, luckily, DNA has a proofreading mechanism. It's kind of like Grammarly, where it goes through it to make sure that everything is right and that the A's and the G's have found their complements. In other words, an A will find a T and a G with a C and vice versa. So this proofreading repairs are also part of the tools, these enzymes, and they lower this rate to about 1 per billion. Now it's been estimated that we have maybe 3.2 billion. So in a sense, someone might have in their DNA, more than likely has at least 3 errors, 3 mutations, and likely it's not going to make a significant change in their genome or what they appear to be or their functionality. DNA has also been noted to be considered to be a carcinogen. That's been damaged by these ultraviolet light rays. I think I said that wrong, but ultraviolet light rays can cause changes, mutations, and when that happens, or chemicals in the environment, we call these products carcinogens. That's probably a better way of saying that. All right, that's all for now, and that's 12.2 on DNA replication and accuracy and repair. Peace.